Morning, everybody. Thanks for making it out so early. So we're going to start the session. It's from the Genome Editor's desk, Rewriting Mutant CFTR using CRISPR and other technologies. We have 5 really interesting talks. Before that, my name is Sriram. I am an assistant professor at Nationwide Children's Hospital, and my co-moderator is Greg Newby. And these are the talks in the session. And so the talks are going to be 15 minutes long, and we have 8 minutes for questions. So if you have questions, please come to the mic to ask your questions. And then there's also the option to enter in the questions online. The first speaker for the session is going to be Karen Kahnke from Nationwide Children's Hospital.
Good morning, everyone. I'm Karen Kahnke. First, I'd like to thank the chairs for selecting this work to be presented today. Today I'm talking about single-stranded DNA with internal base modifications that mediates highly efficient gene insertion in airway basal cells. I have no disclosures. So how do we make single-stranded DNA templates more efficient at gene insertion? I hope to answer that question and a few others, like why do we need single-stranded DNA templates? And can chemical modifications with internal base modifications improve gene insertion? And also how these chemical modifications work. We're all here because we want to find a durable cure for CF. In 1938, the CF was first described, and early treatment focused on treating symptoms and clearing bacterial infections. In 1989, the CF gene was identified, and since then, CFTR modulators have brought much relief to CF patients, especially those with the most common mutations. But modulators must be taken daily. Gene-correcting airway stem cells poses a durable cure for CF. Stem cells give rise to all the other airway cells. If these stem cells are gene-corrected, they will then differentiate into the other epithelial cells in the airway cells. And this poses a potential durable cure for CF. So how do we get a corrected template into the genome? We can use a Cas9. A Cas9 is a bacterial nuclease, along with a guide RNA to induce a double-stranded break in the genome. The cell's internal repair mechanisms then will repair this double-stranded break, either by a pathway called non-homologous end joining, or NHEJ. NHEJ inserts or deletes random nucleotides to repair the double-stranded break. These random insertions and deletions, however, can disrupt a gene. The other pathway is HDR, or homology-directed repair. This pathway uses a template with homology arms, or sequences that are the same as the double-stranded break site, and it will insert this template into the double-stranded break, repairing it. Adeno-associated virus can carry such a template. AAV is efficient at gene insertion, and it's good at evading the innate immune system. So compared to other options, how much more efficient is AAV? This work in 2019 compared AAV-based templates against adenovirus, lentivirus, and single-stranded DNA templates. And their data shows that the AAV had higher HDR than the other delivery methods. So if AAV is so efficient, why are we interested in other alternatives for AAV? One reason is that AAV is limited in its packaging capacity. It's limited to about 4,800 base pairs, and many genetic disorders, including CF, involve long genes that don't fit into one AAV. So what's an alternative? As I mentioned earlier, single-stranded DNA can be used to deliver a correction template. Single-stranded DNA is vulnerable to nuclease degradation, however. Previous work have used chemical modifications on the ends of single-stranded DNA. One such modification is a phosphorothioate chemical modification for a base. And their data showed that modifying the 3 bases on each end with a chemical modification improves gene insertion over the single-stranded DNA that is unmodified. So why stop with just the end modification? we asked, can we make single-stranded DNA more efficient? So our hypothesis is modifying the internal bases of single-stranded DNA improves gene insertion. And to test this, we started with the previous knowledge of the N-modified single-stranded DNA and the phosphorothioate modified base. There's also a commercially available modification called a 2'-O-methyl, which I'll talk about later. So we started with the three modifications on the ends, but we also included some of the internal bases with the chemical modification of phosphorothioate. To see if these modifications were compatible with HDR, we used an F508del template that was previously described by Dr. Vaidyanathan in his previous work, and a Cas9 and chemical modified guide RNA. As a comparison, we used an AAV template that was also previously described by Dr. Vaidyanathan in his work as a comparison and a positive control. We used a DNA PKCS inhibitor to block the NHEJ pathway as well. After gene editing, we worked with the airway cells. After gene editing the airway cells, we PCR amplified the target sequence. We analyzed the results with ICE and reported the results with percentage of alleles that had HDR. Since we didn't have a basis to work, we randomly selected a template to start with. So we started with modifying every sixth base. And we also started with an every third base modification template. And I don't know if you can see that. So as you can see with the data, that the N-modified single-stranded DNA actually did have higher modifications or HDR than the unmodified. And our every sixth base modified template had an even more significantly higher HDR over the N-modified single-stranded DNA. We also found that modifying too many bases actually reduced gene insertion, as we found with the every third modified base template. To check for toxicity, we looked at cell count of the airway basal cells, and we found that the single-stranded DNA did not reduce cell count compared to the unedited mock cells. We're calling single-stranded DNA that with internal chemical modifications that enhance HDR, enhance single-stranded DNA, or ES-DNA. So I've shown you CFTR. We wondered if this worked in other loci. And I've also shown you with the phosphorothioate modification, we wondered if other chemical modifications worked as well. So we used our ES-DNA template to modify a region in HBBs that is associated with sickle cell disease, and also the CCR5 locus, which is associated with HIV infection. And in both of these loci, the ES-DNA templates had higher gene insertion than the N-modified single-stranded DNA. So this is with the phosphorothioate modification. And as I mentioned earlier, there is a 2'-O-methyl modification that's commercially available that we wanted to try. We designed an ES-DNA template with the 2'-O-methyl modification and found that it also increased gene insertion over the unmodified single-stranded DNA. There are also chemical modifications from commercial vendors, and we wanted to see how ES-DNA compared. IDT has an Alt-R HDR modification that's available for single-stranded DNA. It is a proprietary modification on each end, as well as the 3 bases on the end that is modified with the phosphorothioate bases. And when we compared that to our ES-DNA templates, we saw that both gave higher gene insertion over the Alt-R HDR template in both the CFTR locus and the HBB locus and airway basal cells. So I've shown you that it increases gene insertion. Do ES-DNA corrected cells restore CFTR function? To test this, we corrected the F508del mutation in airway basal cells, or airway stem cells from people with CF. We used both the ES-DNA templates. And after we corrected these cells, we grew them on air-liquid interface to create epithelial sheets. And with the help of our friends from OSU, we sent the samples off to have CFTR function measured by electrophysiology. This data shows that a sample using tracing, where our esDNA templates, the cells corrected with the esDNA templates, responded to forskolin with an increase in the short circuit current, and also responded to the selective CFTR inhibitor by a decrease response. Forskolin is an active gated channel activator. And so this data shows that CFTR function was restored with our templates for the esDNA when compared to the uncorrected cells. And it also they gave between 30 to 60% a gene correction compared to the non-CF controls, and this is above the 20% that's considered therapeutically relevant. We also wondered if this worked in other cell types besides airway basal cells. So we used the esDNA templates to correct the CFTR locus in CD34 positive hematopoietic cells, human umbilical vein epithelial cells, and in T cells. And we found that in all three of these cell types that the esDNA templates did increase gene insertion in these cell types. And this is useful for non-lung cell therapies, so that's relevant. We also worked with the Hester lab, and we used the esDNA templates to correct induced pluripotent stem cells. And surprisingly, we found that the unmodified single-stranded DNA gave the higher gene insertion, significantly higher than the esDNA templates. And this made us wonder then, what is different about iPSCs? Our investigation led us to a recent paper on 3' repair exonuclease 1, or TREX1. And in that paper, they showed that TREX1 limits single-stranded DNA-based gene editing. Our further investigation then, we found that iPSCs do not express TREX1, as seen in this Western blot. But other cell types do, like our airway basal cells. So to continue our investigation on TREX1, we generated TREX1 knockout cells and then used our template, esDNA template, as well as the unmodified template for gene insertion for the TREX1 knockout cells, and found that the single-stranded DNA that was unmodified gave us a comparable gene insertion to the esDNA template with the TREX1 knockout cells. And so in summary, I've shown you that modifying internal bases improves gene insertion with single-stranded DNA, and that correcting using esDNA templates restores CFTR function, and modifications protect single-stranded DNA from TREX1. I would like to thank our wonderful lab at the Vaidyanathan lab for their support and help, and especially Dr. Vaidyanathan for his support in my research career and in trusting me with this project, and our collaborators at Nationwide and at OSU, and especially for our financial support, like the Cure CF Columbus, and Nationwide, and the NIH, and then Cystic Fibrosis Foundation. And with that, I would like to also, when we finish with questions, if you have further questions, my poster is 273, and I'll do my best to answer any questions that you might have.
Please go ahead.
Hi, very nice talk.
Thank you.
You said this, I missed it, but do the modifications affect immune stimulation by the sensitivities relative to modified?
I didn't show that data here, but we did, well, as far as toxicity, I showed that data that it doesn't reduce the cell count. And I know that we did some tests with the p53 response and p21 response, and didn't see a significant increase in that response. So,
Thank you.
okay.
And we haven't measured interferon responses.
Interferon. Okay, that's interferon.
Hi, Karen. Really nice talk. Since these are presumably chemically synthesized, I was wondering if you could comment on the size limits.
Yes, so single-stranded DNA is readily available in under 200 base pairs, and these then would be short single-stranded DNA. Theoretically, single-stranded DNA can be made longer, and it is available, but it's not scalable for research at this time, at least feasible for us anyway. But theoretically, then that would be an unlimited size. I know we have a template that we purchased that was, I think, over 2,000 base pairs, or something like that. But yeah, that is, I know I mentioned that with the limitation with AAV. So theoretically, single-stranded DNA would be, could be made longer. And that is something that I am working on for future directions.
Very cool. Thank you.
CFTR. So very good talk. I saw you mention the modified low iPSC cell.
Mm-hmm.
The actual function after the presentation.
Okay, I think I understand your question. Are you asking if we checked for function after we corrected iPSCs? No, we did not check function for iPSCs. We only did that for airway basal cells, but that is something that we would continue to do if we looked into this, as well as we didn't check that for the blood cells or T cells as well.
I had one question as well. It was really convincing, the data you showed, that the effect was mediated through TREX1. I was wondering about whether TREX1 is known to be a 5' to 3' or 3' to 5' exonuclease, and if you know that, then maybe do you think it'll be possible to tailor your modifications just to that end and see if it would have the same effect as modifying all the way through?
Good question. Yes, I believe that's a 3' to 5' exonuclease. And we did actually check that. I don't have that data shown in this presentation, but we did have a template that we did just on that end, 20 bases in. We've also done a 3' spacer on that end, and did not show improved over the every 6 modified base template.
Thank you. We're not seeing any questions online. Remember, you can always submit them online as well. Thanks so much. Let's
Okay.
get another round of applause for Karen. Thank you. Next is Ariel Esperas, and let's see, right here. Thank you, talking about virus-like particles. Welcome.
Hello. Awesome. Hi, I'm Ariel. I'd like to thank the moderators for this wonderful opportunity to share our work in developing virus-like particles as a way to deliver our editing machinery to our primary HBE cells. I have nothing to disclose related to this presentation. So VLPs are made of viral proteins but lack viral genetic material. As a result, they're unable to replicate, and they do not integrate into the genome. They allow for a broad range of cargoes, including Cas9 guide RNP complexes, as well as nucleic acids and proteins. The VLPs that we're using in this study are delivering primarily Cas9 guide RNA RNP complexes, which limit the cell's exposure to these editing enzymes. And as a result, they tend to have less off-target editing and tend to be less toxic to the cells. The VLPs that we're using are based off of the Bankscoda et al. 2022 Cell paper published by David Liu's lab. And a key feature of those VLPs are these Cas9 GAG fusion proteins, where the Cas9 is linked to the MMLV GAG by a cleavable peptide linker that gets cleaved during VLP maturation. I think it's worthwhile to, again, highlight the key differences between the VLPs that we're using to other common viral vectors. So again, these VLPs that we're using have no genome, and they are directly delivering Cas9 as RNP complexes. So this means that they're non-integrating, and they tend to have the most transient exposure to Cas9 relative to these other cargoes. So we're interested in the gene editing team in these VLPs because delivery remains a challenge both clinically and for our in vitro studies. In particular, we have difficulty delivering to our primary HBE cells, and these are our gold standard model, and unfortunately there are limited commercially available options to deliver to them. We believe that VLPs would be a gentle way to deliver our Cas9 guide RNA RNP complexes to these cells to affect cutting. So generating these VLPs are really straightforward. You just transfect these HEC293 T cells with these 4 plasmids, which encode for key components of these VLPs. After 48 hours, you harvest them from the supernatant and clarify them to remove large cellular contaminants using a PVDF filter. You can then concentrate these VLPs using an ultracentrifuge and store them in the -80. To assess for our VLP's cutting efficiency, we use a T7 endonuclease assay. To give you a refresher of this assay, when you edit a population of cells, only a fraction of the cells get edited. So when you PCR amplify the target locus, you'll get some amplicons with the mutation and some amplicons without the mutation. If you denature and then re-anneal, you'll get some DNA species with a mismatch, which the T7 endonuclease can detect and cut at. And so if you run these out on a gel, you'll get 3 bands when there's been cutting. You could also run it through a bioanalyzer, where you can measure the intensity of those bands. And so we calculate percent editing to be the molarity of the largest fragment divided by the sum of the wild-type band and the largest fragment. It's worthwhile noting that this is an efficient tool to calculate the editing efficiencies between samples within an experiment, but to get a more accurate reading of editing efficiency, we would need to use sequencing approaches, which we did not do in this study. So after validating that we can generate VLPs to cut HEC293 T cells, we wanted to see if our VLPs can edit CFTR in 16 HBE cells. So we generated VLPs targeting CFTR exon 12, and then we seeded 16 HBE cells. And then the following day, in a 48-well addition, the following day we transduced with VLPs. And then 72 hours later, we extracted DNA and ran our T7 bioanalyzer assay. For our positive control, we used VLPs targeting the HEC3 site. These are the same VLPs that were published in the Banks-Goda et al. 2022 Cell paper. And for our negative control, we generated VLPs where we replaced the plasmid encoding for the Cas9 MMLV GAG fusion protein with the GFP expression plasmid. So these are the bioanalyzer traces from these initial experiments. You can see we can get about 60% editing efficiency with our VLPs targeting CFTR exon 12. To orient you to these bioanalyzer traces, the blue arrowheads correspond to the wild-type bands, and the red arrowheads correspond to the T7 products. So following these experiments, we were curious to see if our VLPs can edit primary HBE cells. And so initially we wanted to test if these VLPs can edit undifferentiated primary HBEs. So to do this experiment, we seeded our HBEs in T25s, and then transduced with VLPs delivering our Cas9 guide RNA RNP complexes targeting CFTR exon 12. 72 hours later, we extracted DNA and ran our bioanalyzer assay. On the top right, you can see that we can get about 60% editing efficiency with VLPs targeting CFTR exon 12 in undifferentiated HBEs. We also saw that there was limited amounts of toxicity with this treatment, especially relative to our negative control, which was an AdGFP adenovirus delivering GFP. So following these results, we were curious to see if we can use these VLPs to edit our differentiated primary HBEs at ALI. So to do this, we differentiated our primary HBEs at ALI using our Vertex differentiation media for 30 days, after which we transduced with VLPs, again delivering our Cas9 guide RNA RNP complexes targeting exon 12. 72 hours later, we extracted the DNA and ran our T7 bioanalyzer assay. So you can see we can get about 30% editing on differentiated HBEs at ALI. We did have to do either an EGTA pre-treatment or a sodium chloride co-treatment to facilitate access of the VLPs to the primary HBEs receptors, to the receptors of the VLPs and the primary HBE cells. I would like to give a plug to Ashley Cooney, who presented on using sodium chloride co-treatments to improve transduction efficiency. She's presenting on this on a poster later, and I recommend you go check it out. So following these experiments, we wanted to see if we can improve our editing efficiency even further. One way we thought of doing that was perhaps doing multiple VLP transductions to see if we can mediate an improvement in the editing efficiency. So to do this, we differentiated our primary HBEs at ALI for 30 days, and after which we did multiple rounds of VLP transductions, allowing for a couple days of recovery in between transductions. We then extracted DNA and ran our T7 bioanalyzer, T7 assay. Here's a gel of the T7 products from that experiment. You can see that increasing the number of transductions does in fact increase the amount of editing. This suggests to us that at any given round of VLP transductions, a subset of the population, a different subset of the population is available for editing. You'll also note that the sodium chloride co-treatment had higher editing than the EGTA pretreatment. It's worthwhile also noting that we did have some toxicity with this sodium chloride co-treatment, so for the rest of the talk, we are going to primarily present data where we use an EGTA pretreatment to facilitate access of the VLP's receptors. We're still optimizing our sodium chloride co-treatment protocol. So following these results, we were curious to see if we can use VLPs to mediate a targeted super exon insertion into intron 1 in differentiated primary HBEs. So a super exon is a partial cDNA of CFTR into, in this case, intron 1 in the endogenous CFTR locus to take advantage of the native regulatory elements of CFTR. A super exon inserted into intron 1 could potentially provide benefit for over 99% of people with CF in the United States. A particular of this super exon that we're using in this study has a homology arm that spans CFTR exon 1 and its minimal promoter. So in addition to acting as our HDR donor template, it could also have some functionality as an expression construct. So we were interested to see if these VLPs can mediate a targeted super exon insertion in differentiated primary HBEs. So we differentiated our HBEs at ALI using the Vertex differentiation media for 30 days, after which we transduced with an adenovirus delivering our super exon CFTR HDR donor template, coding for CFTR exon 2 to 27. After a couple days of recovery, we then transduced the cells again with VLPs delivering our Cas9 guide RNA RNP complexes targeting CFTR intron 1. And then a week later, we ran our electrophysiology assays and junction PCR to confirm targeted super exon insertion. So here is the gel of the junction PCR showing you that we can get targeted super exon insertion in differentiated HBEs in conditions that had both the super exon HDR donor template delivered as well as the Cas9 guide RNP complexes. So we were then curious to see if these correlated with CFTR function. So we ran our equivalent current assay, and you can see that in conditions that received both adenovirus plus VLPs, we were able to get CFTR functional rescue. It is worthwhile noting that the adenovirus alone also provided CFTR functional rescue, but I hope you can appreciate that we got significantly more CFTR function from the adenovirus plus VLP conditions. So following this, we were curious to see if we can improve the system further. Something that we were talking about was potentially making an all-in-one particle where we can package our HDR donor template in addition to our Cas9 guide RNP complexes. As a proof of concept for this idea, we first wanted to see if we can package our HDR donor templates in VLPs. And so as I had mentioned previously, VLPs can can package a broad range of cargoes, and so we've been primarily using Cas9 guide RNP VLPs. So we wanted to see if we can generate VLPs that packaged our super exon HDR donor templates. So we generated VLPs that did that. It was able to package our HDR donor template as an RNA by introducing a packaging signal in our construct. So when we co-transduced these two VLPs and 16 HBEs, we were pleased to see that we can get targeted insertion of our VLP super exons into intron 1 in these 16 HBE cells. We then were curious to see if these insertions were functional, and so we ran our equivalent current assay with these VLPs, and we were able to detect CFTR function in conditions that received both the super exon HDR donor template and our VLP Cas9 RNP complexes. So to summarize the work that we've presented in this talk, we were able to show that we can generate VLPs that can efficiently edit 16 HBEs in our primary HBE cells. We can show that VLPs can mediate targeted insertion of our super exons in differentiated primary HBEs, and that these differentiated primary HBEs can tolerate multiple rounds of VLP transductions and achieve higher levels of editing. And we've shown you our initial proof of concept experiment that we can package an HDR template in these VLPs, paving the way for potentially making an all-in-one particle at some point in the future. I'd like to acknowledge the CFFT lab. In particular, I want to acknowledge my boss, Hilary Valley, and the gene editing team, as well as the rest of the members of the team that developed our super exon constructs. I also want to thank the EPICS team that did the functional assays, and I also want to thank Kevin, Cal, and John, and Martin for their guidance and advice. And I'll take any questions. Hello.
Hi. That was a great talk. Thank you. And apologies if I missed this, but I wonder if you've characterized your VLPs at all in terms of size, and I wonder if you have, if that changes depending on how much cargo you load.
We haven't done that quite yet, but that's something that we could potentially be doing in the future. We've just been seeing if they were able to induce the editing. This is kind of still a new project, so we're still working on characterization.
Hi, this is Jim Poole from Toronto. So it's a very nice talk. Looks like it's just like a virus. Why you need to use SORD or EGTA to break the tight junction? Is it possible to design that you can go from the top, not from the basolateral side?
So we've done both with or without these pretreatments, and we weren't able to get editing without these treatments to disrupt the tight junctions. We've also done basolateral transductions, and we got more editing when we did these co-treatments than that route.
Great talk, thanks. For your main super exon strategy, how do you suppose to get homology repair to work in the differentiated HBs?
So we think that there is some level of HDR that is happening. I mean, it's clear that it was getting inserted in these differentiated HBEs, but we are trying different strategies to improve our insertion frequency. We are currently developing new approaches, including a homology-mediated end joining strategy to facilitate improvements in the insertion. But again, our lab was really focused on developing the super exons, and we just needed a way to test them in primary HBs, and so we were really delivery agnostic. So, in terms of getting them in there, I can hear you.
So, my question is, did you do a dose response with the DLPs, or what sort of MOI or
We haven't specifically quantified the amount of VLPs thus far, but we have done dose responses within each batch. We give when we concentrate about 1,000x using the ultracentrifuge, we dose between 1 and 25 microliters, and we get a maximum editing efficiency about 60-70% with our maximum dose. Depending on the site, sometimes it plateaus earlier, and there is some variability between batches. But that is something that we're thinking about doing in the future after we finalize our strategy.
Very nice work. So question I have with respect to the editing directly in the well-differentiated cultures. So since HDR requires cells to be in a state of division for the homology-directed repair, do you have a sense in your ALDI culture in terms of what your level of proliferating cells is?
So we've done some flow analysis to look at proliferating cells. There is some proliferation that's happening. I don't have the exact numbers I don't think we're still working on getting better read on how often these cells are proliferating. But it does happen at low levels, at least to facilitate this HDR.
Question for me too. I thought that was really nice how you could achieve the packaging and insertion from the RNA packaged in the VLP. I was curious if you have any information on how and when it's reverse transcribed and what the efficiency of reverse transcription from the RNA to the DNA would be.
So I think it takes about 12 So when we did the co-transductions of the VLPs, we also staggered them by about 12 hours to give the reverse transcriptase enough time to generate the double-stranded DNA. We don't have a sense of the reverse transcription activity efficiency quite yet.
Thank you. Let's check online. Nothing online yet, so thank you very much.
The next speaker is Dr. Alexandra Piotrowski-Daspit from the University of Michigan.
Wonderful. Hi, good morning, everyone. First, I definitely want to thank the organizers, Jerome and Greg, for inviting me to share some of our work today. So my name is Alex Piotrowski-Daspit. I'm an assistant professor at the University of Michigan in the Department of Biomedical Engineering and Internal Medicine, Adult Pulmonary and Critical Care. And before I begin, I just want to give a little disclaimer. There's actually no gene editing in my talk content today, but that's where we're heading. And I'm still hoping that I can convince you that it's worth your while. So my talk today is titled Polymeric Nanoparticles for In Utero Gene Delivery in Non-Human Primates. These are my disclosures. I'm a co-founder of Xanadu Bio. So just to begin, what is fetal medicine? So this is a branch of medicine that really deals with the health concerns of both the mother and the fetus before, during, and immediately after pregnancy, including diagnosis and also treatment. And what about in utero gene therapy or gene editing or ex vivo cell therapies of genetically modified cells? So these are all sort of very viable options to potentially correct monogenetic disorders. And there's several advantages to potentially doing this sort of prenatally as opposed to postnatally, right? So for example, you might be able to correct irreversible tissue damage. So in the context of CF, for example, potentially correcting meconium ileus or preventing that. And then also the ability to sort of take advantage of sort of the privileged environment in utero, right? And potentially deliver to multiple organs that are impacted by the disease. And so advances in utero gene therapy technologies are also coupled with significant advances in prenatal testing. So in this figure, I'm just sort of outlining sort of the human gestational period and also some of the common prenatal testing that's done. And really one that I want to point out is cell-free fetal DNA screening. It's really a non-invasive prenatal testing. It's done using a simple maternal blood draw. I actually did this myself last month for my third baby. And it's now covered by insurance. And it can tell you a wealth of information, and sort of the amount of information that we can get from this sort of early test that can be done as early as 9-10 weeks is really valuable. So we can diagnose earlier and earlier, and theoretically also be able to treat earlier and earlier. Of course, there are very important considerations for fetal gene therapies, and I just want to sort of outline some of the key advantages, but also the concerns, right? So advantages, sort of as I mentioned, sort of improved biodistribution and that ability to potentially reach multiple organs. There's an immature immune system, so you might not have the same sort of clearance barriers, for example, that you have in adults. You can take advantage of a smaller dose to volume ratio. And as I mentioned earlier too, you can really intervene potentially early in pathology and hopefully prevent a lot of disease manifestations. Of course, concerns include maternal-fetal safety. You absolutely want to prevent any adverse developmental effects. And then, of course, also, especially with gene editing therapeutics, you want to avoid any risk of effect on germline cells. So these are all things to keep in mind as this field continues to move forward. So shifting gears a little bit now, so I told you I'd talk about nanoparticles for in utero therapy. So why nanoparticles? So in our lab, we're interested in delivering a variety of different nucleic acid therapies, and these come in all sort of shapes and sizes, as I'm outlining here. But these are often fragile and subject to degradation if not somehow protected. And so we're interested in using polymeric nanoparticles to both protect the cargo and ideally sort of direct the cargo where it needs to go in the body. So why nanoparticles in utero? Well, we think that these can be pretty safe, and there's lots of potential routes of administration. So I'll tell you in a little bit, we administered nanoparticles in non-human primates into the portal vein. But previously, work by my former colleague demonstrated sort of pretty safe gene editing of beta thalassemia using polylactic coglycolic acid, or PLGA nanoparticles. And here on the bottom, I'm just showing you some biodistribution results. So these were administered in utero in mice at various gestational ages. And what I'm showing you here is sort of post-delivery into the uterus, so you can see sort of widespread fluorescence here. And then post-delivery, you don't see anything in the mom, which is also important, right? So the idea is to treat just the fetus and not the mom. And then here, when you look specifically at individual organs within these fetuses, you can see sort of nice broad spread delivery. And in this context, sort of the target organ was the liver, but you also see sort of accumulation of our nanoparticles in the lung and the gut. And so we thought that continuing with polymeric nanoparticles might be a viable option. So in addition to the PLGA materials that I just described, we're also really interested in sort of further expanding our repertoire of polymeric materials. And so I just want to take a brief moment to describe our sort of newer library of vehicles composed of polyamine coesters, or PACEs, as we like to call them. And what I hope to illustrate with this figure here really is that these are highly tunable, right? So we can modify the polymer chemistry by adjusting the monomer composition. We can tune the sort of polymer chain length by adjusting the polymerization time of our synthesis. And we can also formulate these into various types of nanoparticles using various formulation strategies. And what I also want to point out is I'm showing the structure here of our classic polymer, as we like to call it, and some key features of these materials are that they have a hydrophobic domain here. This sort of helps with stability and reducing cytotoxicity. But really for nucleic acid delivery, what's important is this tertiary amine group here, which provides a mild cationic charge, which helps with increasing nucleic acid encapsulation, but it's also not so cationic to induce toxicity. And then we can also, by varying monomer composition, we can also alter the physical properties of these materials. So really, we've been able to generate a library of materials, and we're continuing to test them in various contexts. So just a summary of what I just described. So we've shown that these materials are also biodegradable. I mentioned that they have low toxicity, high nucleic acid loading, and really they're very versatile, and we've shown them to be successful in a variety of contexts, both in vitro and in vivo, for the delivery of a wide range of nucleic acids, including siRNA, PNA, peptide nucleic acids, plasmid DNA, mRNA, et cetera. And so what's also been really exciting about some of these materials is that, at least in adult mice, we've seen pretty broad biodistribution. So for example, with one of our favorite materials, a pegylated version of our PACE polymer, we see nice distribution to the lungs, but then also the GI tract, as well as other organs, right? So it's not a perfect system, but we're excited to be able to potentially target multiple organs that are affected by CF in particular. So all of the studies thus far that I've shown you were done in mice. So we're of course really interested in how these results translate to larger animal species. And so in order to do this, we partnered with Alice Tarantal at the UC Davis National Primate Center to try some of our materials in utero in non-human primates, so rhesus monkeys in particular. And so one of the first experiments that we did was looking at biodistribution of two different types of polymeric nanoparticles. So first, PLJ, which I showed you earlier. So this is a commercially available material that is used a lot, sort of as a standard in the lab, if you will, and then one of our newer materials. We ended up delivering these intraportally in the early second trimester transfer under ultrasound guidance. And just for reference, rhesus monkeys have a gestational period of about 165 days. In humans, it's about 280 days. But there's a lot of similarities between rhesus monkeys and humans in terms of development, and usually there's a single fetus, et cetera. And on the bottom here, I'm just showing you some of the characteristics of these different nanoparticles that we delivered. And I should also mention, so our first study was really just to assess biodistribution. So we're assessing diluted nanoparticles. And then the sort of schematic on the right here is just kind of showing you our workflow. So we formulated our nanoparticles, sent them to our collaborators at UC Davis, who administered them under ultrasound guidance via the portal vein. They harvested the organs, sent them back to us, and we did a lot of sectioning and staining and imaging, and then we have a custom quantitative microscopy MATLAB code that we use to analyze some of the images. So I'm excited to show you some of this data. So first, I'll show you the PLGA data. So this is that sort of commercially available polymer that was successfully used for in utero gene editing in a mouse model in our lab. And so hopefully what you can see here is that, and sorry, I should have mentioned, the first image is maybe a little bit sensitive, but you can see here that primarily we see signal in the fetal liver and the spleen, kind of very similar to what we see in the mouse, and particularly in adult mice. So this polymer tends to accumulate in the liver and the spleen and doesn't really have very widespread distribution. So as you can see, there's not very much signal in the lung, the GI tract, or other organs. And then, sorry, this is maybe a bit of a busy slide, but what we did next was really sort of delve into each individual tissue and sort of work on sort of robustly quantifying the fluorescence signal within each of these. So what I'm showing you here, the top panel, so the top and third panels are tissues treated with PLGA nanoparticles, and then the bottom, and then the second and fourth panel are control tissues for reference. So, and hopefully you can see some of the examples of the nanoparticle signal. And so what we did was we quantified the signal in bulk. So panel B here is showing sort of the aggregate quantification through all of all three fetuses combined. And then panel C is showing you the breakdown per fetus. And so you can see that there's, again, primarily accumulation in the spleen, liver, and omentum, and maybe a little bit of signal in some additional organs. And so if we quantify the total number of extrahepatic organs that are positive for nanoparticles, we see about 7 or so. And then what's more exciting to us, though, is this PACE-PEG data. So again, apologies for the sensitive image here, but you can see that there's much more widespread distribution. But also importantly, we don't see a signal in the placenta, right? And the same was true for the PLGA. Here we have reduced signal in the liver and the spleen, but a nice signal in the GI tract, kidneys, and the lung, which we're really excited about. And here's sort of the identical quantification of this new material. So again, looking at images and then the quantification of the images below. So we still have some signal in the spleen and liver, but we really have widespread distribution, including into the GI tract and the lung, which is really exciting, right? And so if we quantify the number of extrahepatic organs that are positive for nanoparticles, it's about 13. So really exciting. And then as a follow-up experiment, we just delivered EGFP mRNA with our PACE nanoparticles, also to try to understand sort of what's the relationship between actually sort of delivery or accumulation in an organ and then also transfection efficiency, right? So actually delivering a nucleic acid cargo and having it take on its desired function are sort of two different problems. But we did see that there are a number of organs that did fluoresce green. So I'm showing you the liver because that was actually the most pronounced. But we did see about 7 extrahepatic organs in our initial analysis that showed a positive signal. So this was exciting, including the lungs and the GI tract. And then I mentioned this when we were looking at the whole organ fluorescence imaging, but an important check too was to make sure that there was no crossover to the placenta, and there wasn't in either case, right? So PLGA and PACE-PEG look pretty identical to the controls. So hopefully, to summarize, I hope I've convinced you that PLGA and PACE-PEG nanoparticles do successfully distribute to fetal organs after in utero delivery via portal vein in a rhesus monkey. And also that so far what we've seen in terms of biodistribution in the fetal NHP model is that it's consistent with what we see in mice. So PLJ nanoparticles are primarily in the liver and the spleen, whereas our PACE-PEG nanoparticles demonstrate widespread distribution to fetal organs. And also delivery of mRNA encapsulated in these nanoparticles results in successful EGFP expression in fetal organs. And we're really excited to move this forward and look at actual gene therapies and gene editing therapies in particular. So to summarize, there's a lot of people to thank for this. So most of the start of this project was during my postdoc at Yale, so I'm very grateful to my former postdoc mentor, Mark Saltzman, and then also Anna Lin has been a big player in this project, and it was initially funded through the NIH and the Somatic Cell Genome Editing Consortium, and sort of follow-on work now in my lab is funded by the Cystic Fibrosis Foundation. So my student Ariana is going back to these tissues and looking specifically at what are the cell types that we're getting into and sort of trying to extract more data from these precious experiments. We're of course thankful for our other funding sources, and we're always recruiting. So if you're interested, particularly in a postdoc, for example, in my lab, please feel free to contact me. And my poster is $295 if you want to stop by and talk to me later. Thank you very much.
In your gene therapy is always provocative to think about. You didn't see deposition in the placenta. Why not? Wouldn't you may have perhaps seen some? And second of all, did you check the mother to see if it's possible it went through the placenta, transferred across the two circulatory systems, and somehow deposited somewhere in the mother? Because that'll be a big issue if you're going to actually treat both the fetus and the mother.
Absolutely. I didn't show the data, but we did look at the mother, or Alice Tarantal and her team did. They didn't see any signal. And perhaps there's no signal in the placenta because we didn't go through the umbilical vein. We went through the portal vein in the fetus. So, but I agree. I was a little bit surprised, but we saw the same thing in the mouse model. We don't see crossover into the placenta, but thank you.
Very nice talk, Alex. I'm amazed sort of how you continue pushing the boundaries where and how early we can treat sort of with genetic therapies. But one question I have for you is, right now you're obviously testing in utero or in the fetal tissue your biodistribution, but do you think ultimately optimization you have to do all in sort of fetal tissue, or would something like what Dan Sigwood, for example, show, like his showdown piece, would they work also in the fetal tissue? So do you have any understanding of that?
Oh, absolutely, yeah. And so I certainly don't think we should be doing optimization. These experiments are very intensive and very expensive. So no, certainly I think that we're doing more so in mouse models. But absolutely, I think different technologies and Dan sort nanoparticles would be great.
But you're confident that sort of what you see in an adult animal will translate back to the fetal tissue?
So that's part of the sort of motivation for these experiments too, was, you know, not only adults versus fetuses, but also how do results in mouse models translate to non-human primates. And we're also doing studies in pigs, and hopefully if funding works out, in sheep too soon. So we're really interested in sort of how we can correlate these things and hopefully build a roadmap so that we don't have to do extensive testing in large animal models. But so far, results do seem to correlate, but of course, it's early days still. Yeah.
Oh, a wonderful talk.
Thank you.
With the widespread distribution of organs that you're seeing, perhaps from mouse studies, that you know cell types you're getting within each organ?
Yeah, absolutely. So with the PACE-PEG nanoparticles that we're really excited about, so we've looked primarily within the lung and the liver. So in the liver, we're getting more endothelial cells than hepatocytes and Kupffer cells, which is interesting. And in the lung, with the PACE-PEG, we're able to target endothelial cells and epithelial cells, but also some leukocytes.
I just have a quick question in terms of the gestational age.
Sure.
Did you test different gestational ages, or do you know if there's like a critical window of when these lipid nanoparticles will be delivered the best? I'm just wondering in terms of with some of those prenatal tests you were talking about, they rely on the amount of fetal DNA versus maternal DNA, and their sensitivity increases over time. I'm just wondering if there's like a critical window or a difference in that gestational age and where it relates to kind of the time when the sensitivity of those tests is highest?
Yeah, that's a really great question. So a lot of the sort of early testing is, so in the first trimester, there's still a lot of organ development going on, et cetera, and so perhaps that's not the best time. And really only at 10 weeks are you able to test, and that's sort of towards the end of the, or to be able to do the cell-free fetal DNA test, rather. And that's sort of towards the end of the first trimester. So I think that the early second trimester is probably the earliest possibility to do an intervention, which is why we chose that particular time point. But in the non-human primates, of course, we haven't been able to test a wide range of time points, but of course that would be sort of interesting to consider. But my gut feeling would be theoretically the earlier the better. Yeah. Come on.
So my question is, looking at the two different nanoparticles, can you see one nanoparticle having a very live distribution cleaner and spirit, and that one having a much more round distribution? Do you know what is the difference? What makes those other particles be able to spread it?
Yeah, that's a really wonderful question, and something we think a lot about, primarily in the context of our mouse experiments. But what we find is really it's sort of the nanoparticle characteristics seem to dictate in large part the vehicle tropism. So and particularly size and also chemistry, right? So the PLJ nanoparticles don't have that pegylation, which historically, is known to allow sort of longer circulation, more opportunities to reach certain organs. So that's definitely a help. Our PACE PEG nanoparticles are also a lot smaller, and also surface charge. So the PACE PEG nanoparticles are mildly positive. So all of these factors, I'm sure, contribute. But great question. Thank you.
Hi, Alex. I was just
Celeste.
wondering, what is the size limitation for these nanoparticles as far as weight of the mRNA or maybe delivery?
That's a great question. So we've tested CFTR mRNA in another PACE system. And so theoretically with nanoparticles and lipid nanoparticles, you can encapsulate pretty large cargoes, but the downside to that is you can also start to impact the delivery vehicle characteristics, which I was just saying is important for biodistribution. So there's this sort of balance, right? Or there's this careful interplay between what you can load and then how that results in sort of modifying the characteristics. Yeah, thank you.
I have one more question. It kind of goes back to the point that was raised previously, right? What are the properties that sort of allow the nanoparticles to go to different organs? Have you considered, or how are you thinking about the protein corona that might form around different organs? And do you think that it's really dependent on that, and is there a way to modulate it somehow?
Oh, absolutely. So the nanoparticle characteristics will inherently sort of influence the protein corona as well, and we're starting to sort of dig into that deeper. I don't have a great answer for you in the context of this specific presentation, but that's absolutely an important consideration. Awesome. Thank you.
Thanks.
Thank you. Speaking to us next is Lucia Nicosia from the University College Cork. Welcome, Lucia. Talking about adenine base editing of G542X.
Thank you for the introduction. I also hope that everybody's awake by now, including myself, to be honest. So I don't have any disclosures, but I would like to thank the amazing international team who worked on the project that I'm going to present today. Now, I would like to start with a quick recap of the basics, just in case some of you may not be familiar with the field, or in case some of you haven't seen one of these in a while and forgot what a cell looks like, okay? So as gene editors, we work mainly in the nucleus. That is where the DNA is found. Now, for protein-coded genes, an RNA polymerase transcribes the DNA all the way to the termination site into an RNA molecule that then is processed into a messenger RNA that is translated by ribosomes into a protein. But if your termination site is here and you have a premature termination codon upstream, then this translation process is stopped there. So what happens is that two mechanisms are triggered that are called nonsense mediated decay and ribosome quality control. And those will result in the degradation of both the mRNA and the truncated protein that was synthesized so far. So basically, when you have a premature termination codon, you end up having no protein. And that's the case with the mutation I'm working on, that is G542X. So we propose a gene editing strategy to prevent nonsense mediated decay and the ribosome quality control, and to restore the synthesis of the CFTR protein. The gene editing strategy we use is called adenine base editing. So this is our editing machinery. It binds to the mutation, G542X in this case, by complementarity of the spacer, here in light blue. Then you see in blue a nickase domain, and in pink a deaminase domain. So this is called adenine base editor. So it can edit an adenine, and that's the adenine we are interested in, the bottom strand. So the deaminase domain can deaminate the adenine. So what does that mean? It removes the amine group of the adenine, so it leaves us with an inosine. But now this has just created a mismatch because the inosine should be paired to a cytosine. Instead, we have a thymine here. But this is what I find really elegant about this system. Since the cell doesn't know which one between the two nucleotides is the mismatched one, the nickase will nick the non-edited strand, thus creating a gap there, and now we have a single strand break that needs to be repaired. So the DNA polymerase is called, and we'll use the bottom strand as a template to fill in the gap in the top strand. So now you see we have a cytosine there, but this has just created a new variant, G542R. So we are quite literally rewriting a CF-caused mutation, so it's quite fitting with the title of the session. But so why? Why did we choose adenine base editing if we're not restoring the wild-type sequence? So base editing is highly efficient, also in non-replicating cells, so also in fully differentiated cells. It has a minimal risk of insertions or deletions formation that can cause the disruption of the sequence, and it has minimal packaging requirements. So for packaging, we use the EVLPs that Ariel presented 30 minutes ago, but our EVLPs do not have any template. Again, we only need the editor and the gRNA. So they deliver the editor to the cell, then it has some nuclear localization signals that actually allow it to enter the nucleus where the editing will occur. So what type of cells did we do this in? We have access to rectal biopsies of patients that are homozygous G542X, and that I'd like to thank for donating their cells. And so from these tissues, we can retrieve cells that then can be grown in the lab into so-called organoids, intestinal organoids in this case. So organoids mimic an organ, in this case, of course, the intestine. They're extensively used in the field, but just to recap, they're often used as a qualitative assay on CFTR function. So when you have a wild-type CFTR, which would be here in light blue, you have the transport of chloride to the luminal side of the organoids. And now, of course, to ensure that you have the correct balance of salts and water, then water will move in as well. So the organoids will start filling with water and they will swell. If you have G542X, again, remember nonsense mediated decay and so on, you have no protein at all, and then there is no transport of chloride, no transport of water, and the organoids do not swell. So this is a video on time lapse, just to show you exactly what I'm talking about. So these are G542X organoids. You can see the lumen is this inner circle there. Again, there is no CFTR, so they're not swelling. So this is what we started with. We trypsinized these two single cells, and we treated them with the EVLPs. And 7 days later, this is what we saw. So you can clearly see that there was restoration of the swelling property, which is indicative of restoration of CFTR synthesis and function. So we, to answer one of the previous questions, we did perform a dose response. And so we call this multiplicity of transduction rather than MOI for infection, because ultimately these are not infectious particles. And we tested, say here in green, increasing doses of EVLPs, and we looked for the number of organoids that were swelling. So we see an increase in the percentage of swelling organoids with increasing doses of EVLPs. And we also tested a combination of ETI in addition to the EVLPs, and we did not see any significant difference at any of the doses tested at the concentration that we used in this study. Then we also looked at the extent of the swelling, so how much is the area increasing over time. So you see in black is the control G542X organoids. There is no swelling over time. And then you see in green, again, the edited organoids. In orange, edited organoids plus ETI. Once again, no difference between the two. But we were also interested in measuring, quantifying the conductance of chloride mediated by CFTR. So we also grew the organoids as a monolayer on a filter membrane in a liquid-liquid interface, because then this can be mounted on the using chamber that I'm sure most of you are familiar with. So that allows us to quantify the net flow of chloride ions from the basal to the apical side. So this is the result of our using chamber. Now, as most of you know, we are interested in the response to the CFTR-specific inhibitor. So here would be this orange or brownish there. Because we activate CFTR levels above baseline levels using forskolling, but that is not specific to CFTR. So then we're interested in the response only to this inhibitor. So you can see for the first two, those are our negative controls. There is no orange, so we don't have any CFTR-specific chloride conductance. You do see orange here after the editing, and after editing plus ETI, once again, no difference between the two. Then we also added here a wild-type control as reference. And so if you're wondering how much we restored relative to wild-type, that's 9%. So we recovered 9% of wild-type function. But I would like to stress this point now. This result comes from the entire population of cells that are on this filter. We did not perform any enrichment strategy for edited cells. No, it's not a clonal population, it's the total population of edited cells. So after the using chamber, we scrape the cells off the filters to extract DNA and send that for sequencing. And it was quite surprising that we had as little as 2% of DNA editing. So 2% of DNA editing was sufficient to recover 9% of wild-type function. Now, there are some concerns about like adenine base editing or base editing in general. And one of these is the bystander editing. So we were interested in editing this adenine here, position 9, but there in that region, there were other adenines standing by, so the bystanders. So we wanted to make sure that we were only editing our target adenine. So we looked at the other adenines there, and we could not detect really any bystander editing above background levels. And then the other concern that is common to every gene editing approach really is the off-target editing. So what if your guide-RNA binds to another region of the DNA that is highly similar in sequence? Now, there are web tools that allow you to screen and predict potential of target sites. So we checked the top exonic of target site, and once again, we saw no off-target editing above background levels. Now, the $1 million question that we always get asked as gene editors is how much is enough? So how much DNA editing is enough to restore wild-type CFTR function? Now, there are some studies that date back to the early 90s and early 2000s that have tried to answer this question, but they are based on experiments that mix wild-type cells with mutant cells and try to understand what's the percentage of wild-type cells that you need, or they use a gene transfer approach. So what I did here was a little bit of a literature review. I went back to all the papers that were published for like on gene editing for CFTR, and I plotted here the editing efficiencies reported by each strategy, by each study, versus the functional restoration that was achieved. So now I was looking for the ceiling effect that was described by Boucher in 1992. So I plotted an exponential decay curve, and it plateaus at 23%. So it is quite likely that that is the target. About 23% of DNA editing might be enough to restore wild type or near wild type levels of function. Now, so we're not quite there yet. We have a tenth of that, but we have seen how the EVLPs can successfully deliver the editor to patient-derived intestinal organoids. We've seen how as little as 2% of editing to G542R can restore 9% of function relative to wild type with no bystander and no off-target editing. Now, if you're shy, I can meet you later in my poster, number 300. Otherwise, I'm happy to answer any questions now. Thank you.
Interesting talk. Thank you very much. Tom Lynch from Nationwide Children's Hospital. I'm curious about the kind of the technical parameters of designing the guide RNA. How close does it have to be to your site, and how translatable is your approach for this mutation to other premature termination sequence?
So you ask about the guide RNA, right?
Yeah,
Sorry,
designing the guide
yes.
RNA. How translatable is it to other mutations?
So for base editing, the guide RNA needs to be designed on the mutation. And depending on the editor you use, there is an editing window, there is an activity window. The guide RNA is usually 20 nucleotides long. It has to be specific for that. But it also has to have a PAM sequence, so-called. It's like an anchoring sequence for your editor. So there are some limitations there. It also has to have a certain percentage of Gs and Cs for stability, which is not quite common on the CFTR gene. It's super 80 reach, so that is a bit complicated. And not only that, different sites, different loci in the gene of CFTR have different accessibility. So how is that translatable to other mutations? It also depends on the chromatin profile there. Anne Harris characterized this extensively. Now, different loci can be more packaged into the chromatin, so that might make it more difficult to access. And I think in the next talk, we'll see one approach to kind of improve the access there. But yes, it has to be tailored on the mutation.
Hi, nice talk. Do you have any idea how changing a G to an R at 542X changes the function of CFTR, or if it does change the function?
So I should have an extra slide here. Always refer to this study from David Bedwell, which I think dates to 2017. So you're familiar with his work, I'm sure. It's on a read-through, spontaneous read-through. So G542X sometimes you have some read-through to G542R. So in this study, they created a cell line, a Fisher-Rat thyroid cell line with the cDNA, the human cDNA, carrying G542R. So this is 100% of cells with G542R. So you see already in the Western, without the addition of the X809, the ratio band C to B is around 1. So you see how that compares to a wild-type CFTR instead. So it is quite likely that, maturation is lower because we have 1 as a ratio. And then you see the function here. So it's about 25% of wild-type function. So yes, it's not ideal, but the reason why we went with this is because, how do they say it? Perfect is the enemy of good. So in this area, which is 80 reach, it is quite difficult to get any editing. So there are other approaches such as HDR and prime editing, but they also come with other byproducts such as indels or integration of the scaffold. So we went with this strategy because it's quite clean and it gives you something, because people with this mutation at the moment have no treatment.
Thanks, Kai.
Hi, thank you so much for the talk. So I was sort of trying to grapple with the numbers, and I wanted to ask a little bit about, say, if you had done a wild type study and then scraped the filter and tested how many of the cells were positive, are you expecting that you would see something like 20% which would make the 2% that you got be 10% of function? Or like, is it the case that maybe you were elevating the CFTR function far above what you would typically see? I guess that they both seem really interesting, so I was curious.
So the relationship between, say, in this case, the editing and the function is not linear, like we've seen in that graph. I'll show you something else. So it's always from this literature review I'm kind of working on. I looked at the difference even between different tissue types. You see airway versus digestive tract. This is, again, data from studies that have been published in the field. You often see a higher editing efficiency in airway cells compared to the cells from the digestive tract, but the function to editing ratio is much higher in the digestive tract. And now this could be attributed to some repressive transcription factors that are specific to the airway that might ultimately reduce the transcript levels of CFTR. So it is kind of a difficult question to answer. I'm not sure. But
Very interesting. Thank you.
So very nice work. Question, so if you're at 2%, so obviously you want to be able to increase that. Are you going to do that through sort of successive delivery of the VLPs, or what is your proposal going forward?
Yeah, so when I first started this, I was in my second year of my PhD, so my strategy was, let's just add more until we get something. But we have not reached a plateau yet with the EVLPs. So now I'm trying to study that. Of course, transduction efficiency doesn't necessarily equal editing efficiency, so I'm trying to study that, and perhaps adding too much of the EVLPs might be counterproductive. So we'll see that. Another approach could be pseudotyping the EVLPs as well. So they have a glycoprotein. At the moment, we're using VSVG, which has a very broad tropism, but we might change that to a glycoprotein that is specific for a specific cell type. So we are targeting what we want, and we're not losing, let's say, EVLPs on other cells that we're not interested in, for example. We'll see.
Hi, you kind of already answered that with this slide, but I just kind of wanted to ask if there was any analysis in terms of the specific cell types within the organoids that might have been preferentially edited. I guess that's kind of hard to do without doing single cell patch clamp, but was there an enrichment in some of your organoids by cell type when you saw this kind of linear relationship?
So we did not do that. We thought about the single cell measurements for CFTR, but with 2% and without an enrichment strategy, that was not possible. So I'm not sure. But that, of course, is the most interesting part in the end. What did we target? Was it stem cells that then differentiated and we're measuring that? Or did we target also differentiated cells? And then again, what is the receptor that is expressed on those cells? Can it accommodate VSVG? So it will be interesting in the future.
Last question from me also. I think Ariel had shown earlier that the salt treatment improved transduction. Is that something that's adaptable to your culture system as well?
I'm definitely going to try salt. Yeah, I think it's important also for the possible formation of aggregates between the EVLPs. That's what's worrying me as well when I'm adding so much, because the multiplicity of transduction we have is around 10 to the 7 EVLPs per cell, which is a huge number. So hopefully, perhaps adding the salts, if anything, again, the EVLPs might not form these aggregates, which we have seen in a transmission electron microscopy we performed. And so then that should increase perhaps the transduction efficiency as well. Right.
Thank you.
Okay, thank you.
And the last speaker for the session is Colin Hemes from the Broad Institute.
Thanks. Hi, everyone. I'm Colin. I'm a graduate student in the lab of David Liu at the Broad Institute. I'm really happy to be here. Thank you all for coming. I'm glad you're here as well. I want to thank the organizers of this panel for inviting me here, and to the Cystic Fibrosis Foundation for bringing us all together. It's been a really exciting first day so far, and I'm really looking forward to the rest of the conference with all of you. So I'm going to be talking today about our efforts to use prime editing to enable the efficient and precise correction of CFTR F508del in primary airway cells. We've filed a little bit of IP on this work, and that's my only disclosure. Before I get into the science, I want to acknowledge the amazing team of people who made this work possible. I co-led this work alongside Alex Souza, another graduate student in our lab, and working with him has been just utterly amazing. So I can't thank him enough for his collaborative work with this. I also want to thank other people in our group who played a strong role in this study, including Jordan Doman, Kita Oye, and Smriti Pandey. The people in our collaborators groups who also made this possible, Paul McRae, Lele Sumbatroy, Katerina Kohankova, and Philip Karp, as well as Greg Newby, who contributed to this work while he was still a postdoc in David's lab. So I sort of like to think about the history of gene editing technologies as a decades-long attempt to answer this question: what can we do with a nick? CRISPR-Cas9 is a pair of scissors, and we can program it to cut one or both strands of DNA in living cells at a specific location. And with that capability, we have to answer the question, what can we do with it to install precise changes in the genome that could have a therapeutic effect? The first generation of these sorts of technologies were nuclease editors, which Karen described really well in the first talk of this session. You can deliver a nuclease editor or a nickase editor along with a single-stranded DNA donor that encodes the specified edit and gets incorporated into a cell via homology-directed repair. What else can you do with a nick? Well, you can fuse your Cas9 nickase with a deaminase enzyme to make a base editor, and Lucia gave an excellent overview of how base editors work, so I'm not going to go into that in too much detail today. What else could you do with a nick? You could fuse your Cas9 nickase to a reverse transcriptase and use it in conjunction with a guide RNA that encodes a specified edit that you want to install in a particular location. And that's a prime editor, and that's the focus of this talk. There are also some newer technologies that have been described in recent years that pair prime editors with recombinase enzymes to enable precise whole gene integration in living cells at specified locations. So this is a really wide arsenal of gene editing tools that we have at our disposal to create therapeutic edits in human cells. And prime editors occupy a sort of very unique niche within this arsenal that makes them uniquely suited as therapeutic tools. So unlike nuclease editors, prime editors don't rely on double-stranded break intermediates to install their edits. Double-strand breaks can cause the formation of undesired indel byproducts, large genomic rearrangements, and the activation of p53, for example. Unlike base editors, prime editors are capable of installing all small DNA sequence changes, so transition point mutations, transversion point mutations, small insertions, small deletions, and all combinations thereof. And unlike the newer recombinase-based technologies, prime editors can be encoded in an entirely DNA-free system. They don't rely on the co-delivery of a DNA donor, which greatly expands the scope of delivery technologies that can be paired with prime editors for in vivo applications, and also reduces the immunogenicity of the delivery. So these traits of a prime editor, the DSP-independent mechanism, the ability for it to install transitions, transversions, insertions, and deletions, its ability to be encoded as an all-RNA or RNA protein system, and its ability to function in both dividing and non-dividing cells makes it a really useful tool for exploring therapeutic gene editing strategies. So here's a little bit more about how prime editors work. A prime editor at its core consists of a Cas9 nickase fused to a reverse transcriptase that's paired with what we call a prime editing guide RNA, a PEG RNA, which both specifies the location of the edit as well as the sequence of the edit that we are installing at that precise location. The action of a prime editor enzyme results in the formation of a DNA flap on the genome, which with flap equilibration and endogenous DNA repair results in the installation of the edit on both strands of DNA within the cell. So Alex and I were really interested in deploying this system for the correction of CFTR F508del, the predominant genetic cause of cystic fibrosis in patients with CF. And our initial results that using this form of the prime editor for F508del correction were really discouraging. We only got about 0.2% editing at our sort of first attempt at this in a HEC293T model cell line, which was 2 orders of magnitude below our target editing of about 20%, which we had set for ourselves at the outset of this project. This was kind of hard to see. So Alex and I sat down, and we sat down again, and we sat down again, and thought really deeply about the potential rate-limiting steps in the prime editor mechanism that might be limiting our editing efficiency at this location. And we identified a huge number of variables that we could explore that might influence prime editing efficiency. And most of this project involved systematically exploring these variables to gradually increase our editing levels to percentages that could have a therapeutic effect. So we started exploring all of these variables one by one. Right off the bat, we used some recently described prime editor systems that involved engineered Cas9 nicases, 3' extended guide RNAs, and a dominant negative form of MLH1, a DNA mismatch repair protein, that together sort of boosted our editing efficiency about 5-fold. So the Cas9 nicase has enhanced nuclear localization to the nucleus. The EPEG RNAs, as we call them, are a little bit more resistant to exonuclease degradation within the cell, and MLH1-DN helps to inhibit DNA mismatch repair and favors edit incorporation. Once we were at 0.5%, we felt like we had sort of enough of a foothold on editing above the background levels in order to engage in a much broader search of the characteristics of the PEG RNA itself for editing. So we performed many screens of several hundred PEG RNAs, one of which I'm just showing here, to investigate numerous protospacer sequences as well as the characteristics of the edit encoding portion of the PEG RNA to identify the sort of optimal PEG RNA characteristics for this edit. We also investigated numerous Cas9 guide scaffolds that have been described over the years. Once we had sort of pinned down the optimal PEG RNA characteristics, which got us close to 1%, we performed a nicking guide screen. So we co-delivered a single guide RNA that nicks the opposite strand of DNA relative to the PEG RNA and serves to sort of bias DNA mismatch repair in favor of edit incorporation because it's forming a nick in a nearby location to where we're forming the nick for the actual therapeutic edit. And by screening a number of adjacent nicking guide RNAs, we found one that gave us a nice boost in editing without boosting our edit to indel ratio too dramatically, or I should say without decreasing the edit to indel ratio too dramatically. Once we had a nicking guide RNA, we started to investigate strategies for encoding edits that were preferentially incorporated due to their intrinsic evasion of DNA mismatch repair. So here I'm showing the actual sequence of the CFTR F508del locus. The therapeutic edit we're interested in making is that CTT insertion that we see in green. And we investigated the incorporation of translationally silent edits that lead to DNA sequence changes, but not protein sequence changes. These silent edits form little bubbles of DNA mismatch when they're incorporated on one strand of the genomic DNA, which are less effectively recognized by DNA mismatch repair mechanisms. They also function to encode changes in the DNA sequence that prevent the prime editor re-engagement with the protospacer at this location, which helps to reduce the formation of indels. If the prime editor cannot engage with this site, it won't be able to form an indel. And we found one editing strategy, which we call SE2, that gave us about a 10-fold boost in editing efficiency in HEC293T cells. Next, we investigated the reverse transcriptase component of the prime editor. So Smriti Pandey and Jordan Doman in our group used directed evolution to generate reverse transcriptase variants that functioned with higher efficiency in prime editing contexts. These are called the PE6 suite of prime editors. And we found one editor here, PE6C, that gave us about a 1.5x boost in editing in HEC293 T cells. Now, as Lucia mentioned in her talk, we have had this sort of lingering suspicion about the CFTR site that local chromatin context or proteins engaged with the gene could be reducing our ability to engage the prime editor at locations of interest, and that this reduces editing efficiency as well. So what we investigated was a strategy to kind of disrupt local chromatin context, and we did that with the incorporation of a catalytically dead guide RNA. We call it a dsgRNA. This is a guide RNA that is truncated at its 5' end and is capable of binding to, but not cutting, a site of interest adjacent to the site of our therapeutic edit. The dsgRNA seemed to improve editing by about twofold. Here, this is in 16-HBE cells relative to the use of no dsgRNA. So that was really nice to see as well, and we incorporated the dsgRNA into future formulations of our editing strategy. So when all this was said and done, we kind of took a look back at our whole editing approach and saw that all of the improvements that we made over the course of our optimizations are crucial for obtaining high editing efficiency in 16-HBE GE cells. So this is 16-HBE cells that are edited via mRNA nucleofection. We obtain about 50% editing efficiency in these cells. The one component that we found that we could sort of sacrifice in this case was the dominant negative MLH1-DN protein. This was also really nice to see because MLH1-DN co-delivery presents sort of additional challenges to delivery. It's another protein piece that we'd have to worry about when delivering to cells in vivo. And also it could sort of transiently reduce MMR sort of broadly within the cell, and there's worries that that might increase the sort of basal mutation rate of cells, at least transiently. So if we can do away with it, that's good, and it seems like we can do that. We took a look at the purity of our edit, especially in relation to Cas9 nuclease strategies that had been described for F508del correction previously. Whereas the edit to indel ratio for Cas9 nucleases hovers around 1:1, the edit to indel ratio for prime editors was about 4:1. And this was really nice to see because it means that we are preserving unedited ETI treatable alleles with our editing strategy. So if the cells are not edited, they are still F508del and therefore will respond to ETI treatment. Once we had this strategy in hand, we handed off our strategy to Paul McRae's group, who edited primary airway cells from people with CF that were homozygous for F508del. We got about 25% editing. We also performed an experimental off-target nomination method called CircleSeq to identify potential regions within the human genome that were edited off-target with our 3 guide RNAs, the EPEG, the nicking guide, and the dsgRNA. Out of 32 sites that we tested for all 3 guide RNAs, we only found a handful that seemed to edit slightly above untreated controls, and all of these were within intragenic regions or regions of unknown functional consequence. And finally, we tested the efficacy of our edited cells for CFTR function with electrophysiology. And we found that with about 25% bulk editing, we got restoration of CFTR function to levels comparable to treatment with ETI. This was really exciting for us to see. So to conclude, we're sort of interested in pursuing this strategy a little bit further, and we're asking ourselves 3 key questions as we sort of move forward with this work. We learned a lot about how to optimize a prime edit for therapeutic purposes, and so we're asking, can we generalize our optimization approach to other CF-causing alleles? Can we package and deliver our enhanced prime editing strategies using viral, virus-like, or non-viral vectors? The prime editor is really big. We have 3 guides. We might need to incorporate MLH1-DN. That poses a significant challenge for delivery, and so we're exploring lots of different strategies for doing that. And finally, does the functional rescue we see in vitro translate to some sort of functional rescue in vivo? Can we actually edit F508del or another allele in an animal model and demonstrate phenotypic rescue? We're really excited to engage in this work. So that's about it. I want to thank you again for your attention. The people with cystic fibrosis who donated samples for this study, our collaborators, our funding sources, and I'm happy to take questions.
Great, beautiful work. So a question that I have is, you still have maybe a 4-to-1 ratio of your desired edit to the indels. So is there a strategy? I mean, ideally one would want an editing strategy that would be, precise and not introduce. So is there a way of sort of adapting the prime editing strategy so it becomes more specific? In your case, where you're just leaving a mutant sequence is not problematic, perhaps, but I think for other applications, one would want to have something where you don't risk having, in this case, if you have 50% on target desired editing, then you have 10%, 12.5% of indels.
Yeah, absolutely. And it is something that could have a significant sort of consequence even for this edit, because indels generally result in frame shifts, which result in premature stop codons. And so those are much more difficult to address with modulator therapies if we're installing a lot of those. There are some sort of tools in the toolkit that we know tend to reduce indels or increase edit to indel ratio. The translationally silent edits, which I previously mentioned, do so. And we've investigated about 150 more of those since this work and have found some that increase efficiency and also seem to increase edit to indel ratio. The nicking guide plays a really strong role in determining the indel profile of the editing strategy. So the nicking guide creates another nick nearby the nick of the edit, which increases the proclivity for double-strand break formation. So there are strategies such as the PE3B strategy, as it's called, which was described in the original Prime paper, which sort of segregate temporally the formation of those nicks in order to prevent any sort of double-strand break-like intermediate. There are some strategies that we can actually explore for this site that we have done a little bit of limited work on, and re-exploring PE3B strategies for this site might sort of help with that.
Congratulations. This is really a hopeful talk.
Thanks.
I think I have a question regarding the PEG guide RNA design. So you actually try so many of them, and in order to get higher efficiency. I think if you use computer programs to check, you will check the first one you found actually is not very good. You actually switched a little bit. So that means for other mutations, if people want to try prime editing, so they should test many different guide RNAs, PEG guide RNAs, instead of rely on the computer program.
Yeah, so the question is, can we use models, many of which have been described to date, for predicting sort of optimal PEG RNA characteristics for our edit? So after we had sort of optimized everything empirically, we did go back and use some of these models, most notably Deep Prime and Predict, which are two models that exist for this, generated PEG RNAs using those models and then compared them against our current best editing strategy, and we found that they underperformed. We found that some models worked a little bit better than others, some models worked better for some sites than others. So we learned a lot about maybe how to work with those models, and the way I kind of think about them is they're useful, and they're useful for maybe getting a general sense of the spaces you need to explore in PEG RNA parameter space in order to get editing levels to a decent level. But we think that sort of empirical kind of brute force testing of many different PEG RNAs is still sort of a surefire way to get the best possible editing strategy.
So in this case, you need to insert 3 nucleotides.
Yeah.
But for others, maybe point mutations, you just need to change 1. Is this a special case, or you think maybe for point mutations it will be easy to handle?
Yeah, I don't think there's necessarily anything unique about this edit that we're making. It's not necessarily the nature of the insertion that we're trying to make. we have found that this site is strongly recalcitrant to prime editing for reasons we still haven't really pinned down, and that's kind of what necessitated all of the optimization work that we're doing here. But I think the lessons that we learned from optimizing this strategy, we're already applying them to other CF-causing alleles as well as other genetic diseases, and we're finding that the sort of broad lessons learned are still kind of holding true. So we hope that that's useful for the rest of the community if they're interested in using prime editing for therapeutic edits.
Hi, very nice talk and very good progress for the prime editing in the CF cells. So I have a question is, I wonder if you have some Cas9 value and optimization to improve your editing rate. Another question is, can you comments on the prime editing downside and compared to the Cas9 HDR repair and also the newly developed CAST editing? Thanks.
Yeah, so the first question is about, can we compare our prime editing strategy to Cas9 nuclease strategies and what the sorts of outcomes we see are between those? Is that correct?
Yeah.
Yeah. Yeah, so we didn't sort of do a direct comparison of Cas9 nucleases against prime editors in this study. There have been some Cas9 nuclease-based strategies for F508del correction described in the literature in the past. The main sort of feature that we see in those strategies is the approximately 1-to-1 edit to indel ratio. And I'm not sure what exactly the nature of those indels are or how they might compare to the indels that we do see that get formed with our prime editing strategy. We just know that the ratios are substantially different. Lucia.
Amazing talk. Congratulations. So I just meant to ask, apologies if I missed that. What's the rationale on the design of the DAG RNA? Is it based on ATAC-seq data, or is it just a screening of different sites?
Yeah, so the question is, how did we design the dead guide RNA, the DSG RNA? It was all pretty much empirical. There's a handful of papers that have described using a similar strategy for various purposes, epigenetic editors that want to park a Cas9 somewhere without actually cleaving the DNA. It's been used for prime editing applications in a couple of studies, but it was largely just empirical. So we tested a number of DSG RNAs that target upstream or downstream of our therapeutic edit, the positive strand or the negative strand relative to the therapeutic edit. And we found a few with different characteristics that seem to work. So we're still unsure about what the precise rules are for how to engage a DSG RNA in a site. That differs a little bit from, say, the design of a nicking guide RNA, where we tend to have rough rules about how to design a nicking guide RNA, where to park it relative to your therapeutic edit. A DSG RNA seems a little bit more unclear, and so empirically testing a variety of them seems like the best bet for now.
So really cool talk. A lot of other people have edited F508del. There's at least 4 or 5 labs in the room who've done it. And with HDR, they've not had as lower efficiency. So I'm interested in this dead guide RNA. Is this something unique about prime which is causing a problem? Or is it, do you find it with base editing, or do you find other regions where prime works really, really well? I'm just curious about this locus, as we all are.
Yeah, is there something about prime that makes this this site really hard to edit? We think yes, but we're not totally sure why. And the one piece of evidence that we have for this is we did an experiment where we replaced the prime editor with a base editor, but we used exactly the same guide RNA, the PEG RNA, which encodes the protospacer at that site, and we used an adenine base editor. So we're deaminating bases at the F508del protospacer that we also use for prime editing. 97% deamination. So highly, highly efficient. We did find that if we use an ePEG RNA versus a single guide RNA, the editing efficiency does go slightly lower. And that's likely due to the reduced affinity that Cas9 has for 3' extended guide RNAs relative to canonical single guide RNAs. So what we think is that there might be some sort of kinetic landscape that the prime editor has to navigate, in which the prime editor needs to be in a sort of conformationally active and PEG RNA bound conformation in order to interrogate DNA strands. And there might just be something about the local genomic context of CFTR that renders it difficult for the prime editor to do that on its own. And by incorporating a dsgRNA, we're sort of disrupting local chromatin and kind of shifting the equilibrium of that kinetic landscape into a place where there are more genomic sites that the prime editor is capable of interrogating. And so this might be the means by which it improves editing efficiency, but that's sort of pure speculation on my part.
Yeah, no, it'd be interesting to test some of that, or more of that. Have you tried it with an untethered prime editor, so where you don't have them fused?
We did not try that for here. We tried circular guide RNAs with tethered PEG RNAs, but we didn't try an untethered prime editor.
Okay, thank you very much.
Very nice talk. Just continuing on the theme of the local context, is your hypothesis that this is very sequence specific or more generically chromatin structure? And how does this apply to other sites? Your observations apply to other sites and other editors?
Yes. Is it sequence specific or chromatin related? I'd be inclined to think it's both. So it's been mentioned in this session already that CFTR is a very AT-rich gene. And the AT richness of a protospacer sequence is a strong determinant of how effective Cas9 is at actually engaging with that. So in a sort of broad sense, that would make sense that if we're working with an AT-rich sequence, that might be an overall lower editing efficiency for all Cas9-based gene editing strategies. But there is still something interesting about the local genomic context. In our group, we have edited other AT-rich sequences elsewhere in the human genome and haven't necessarily encountered quite the same recalcitrance to editing that we see here at CFTR. So there's probably a sort of interesting mixture of both.